Hi everyone, my name is Justine Masson and I'm doing research in an academic laboratory in Paris in France. I'm going to present you the study we performed in order to understand the consequence of the loss of the protein YIF1B in humans and mice. Our study is a good example of how basic research done by outright biologists opens the way to the understanding of a rare pathology. Basic scientists do research in laboratories using cellular and animal models. Pediatricians receive families with babies displaying developmental delays, and geneticists find that there is a genetic alteration involved. But how a corrupted gene affects human development has to be investigated. And this is why clinicians end up contacting biologists to decipher the underlying cellular mechanisms that link a genetic defect to a disabling pathology. Clinicians from different countries receive young patients. They were all displaying similar neurodevelopmental defects and pathologies associated with central nervous system alterations. Because more than one patient were identified in several families, a genetic aberration was suspected. By sequencing the entirety of the genome of patients and relative blood sample, geneticists identified several mutations in the YIF1B gene in patients and parents. However, only children carrying mutated gene on both alleles were affected. The next step was to investigate how the different mutations identified could affect the expression of YIF1B protein in the tissue of patients. We used culture of fibroblasts from patients and relatives isolated from skin biopsies and demonstrate that the expression of the protein was undetectable in patients. This was suggesting that the lack of YIF1B was responsible for the pathology. We therefore used a genetic model, the YIF1B knockout mice lacking the YIF1B gene. To evaluate visual acuity of mice, they were placed on a platform of a rotating cylinder with stripes, and mice did follow stripes, as you can see with the movement of the head. This experiment allowed us to show that mutant mice have a decrease in their visual performances, which could be attributed to a retinal deficit. To test motor function, we used the rotarod test consisting of a horizontal rotating cylinder where mice are placed. Over time, wild-type mice learn how to stay longer on the cylinder, whereas mutant mice do not improve their performances. These defects observed in mice are similar to some of the clinical features observed in patients. MRI performance patients show that the lack of YIF1B affects brain anatomy. We checked if similar defects were observed in the mutant mouse brain. We found an enlargement of the ventricles and cerebellum hypoplasia in mutant mice, as observed in patients. We further explored cellular alteration by immunostaining experiment in mice brain section and showed that myelination was altered in the cortex and that the number of specific cells was reduced in the cerebellum. YIF1B was shown to be involved in intracellular trafficking and essential for Golgi maintenance. Function of the primary cilium, a specific tiny subcellular compartment, is known to rely on anterograde trafficking from the Golgi. In this study, we demonstrate that YIF1B mutant mice display some ciliopathic-like phenotype, ciliopathy being a group of pathologies in humans in which the function of the cilium is altered. We thus investigate Golgi and primary cilium morphology in the brains of mutant mice and found some alteration, such as disorganization of the Golgi associated with cell death, a change in the size of primary cilium, and disorganization of centrioles complex microtubule-based structure that form the basal bodies required for the formation of cilia. We then explore the ability of patient cells to form primary cilia and show that, as in mice, human cells depleted in YIF1B have defect in ciliogenesis. Our study is a description of a functional deficit linking pathologies associated with Golgi apparatus and primary cilium alteration. We believe that this pathology should be considered as belonging to a larger class of neurodevelopmental diseases in which the function of protein involved in the trafficking towards specific cell membrane compartments are altered. We deeply thank the patients and their family for their participation and the funding that allow us to complete this study.